Go ahead and start. So we'll start right at the very beginning where we began, which was with the fluid compartments. All right. And so just to kind of review this here, we said that we have two fluid compartments. Okay. So we've got our fluid compartments. We have our total body water, and that was further divided into ICF, fluid inside cells. Okay, this is about 70%, so the bulk of our total body water. And then we also had ECF, fluid outside cells, which was that smaller percentage. Of our ECF, we had two further subdivisions, right? We had the plasma, which is really fluid within the vessels. All right, this was about 10% of that 30%. And then the second subdivision of the ECF was the interstitial fluid. This is sometimes called the tissue fluid, by the way. It's a fluid that's around our tissues. Oops. And this was basically about 20%. All right, so fluid around tissues, and this is about 20%. Okay, and so just having a relative understanding of these proportions is more important than actually memorizing those given values, right? So the larger chunk of that total body water is the fluid that's inside cells, 70%. A larger chunk of our ECF is the interstitial fluid, and then the very smallest component of the ECF is going to be about 10%. That's the fluid that's within our vessels or the plasma, okay? All right, let's move on at the bottom here and speak a little bit about homeostasis, which is the second topic we looked at the beginning there. So we had two major components to homeostasis. Negative feedback was one. Okay, and we said that negative feedback were changes that correct our error signal. Correct or turn off the error signal. The second thing we said about negative feedback is that it helps move our regulated variable towards set point. That could be increase or decrease, but the movement is back towards our set point, okay? And then the second more rarer component here was positive feedback. And that was, again, rare, but this was a brief movement of the regulated variable away from set point. Okay, and then it usually feeds into a negative feedback loop, right? So it's going to feed into a larger negative feedback loop. In other words, positive feedback is really a part of negative feedback, but it's just a very brief time where we move away from set point, deliberately so, for a desired outcome. And some of the examples that we gave here were clotting, blood clotting, which I'm actually teaching right now in AMP2, kind of very interesting here. So blood clotting was an example of that, positive feedback. We also see uterine contractions. Right, as two very good examples of positively or potentiating changes of our variables away from set point for a short period of time. Okay.
I have a question about that positive feedback. Would a fever also constitute as a positive feedback because it's the body's way of trying to kill what's ever inside?
A fever is not really a positive feedback. It is a desired raise in temperature, but that's really an inflammatory response. I guess we could consider the fever itself, like the initial part of that inflammatory response, to be positive feedback, but it's really a negative feedback because we don't want body temperature to exceed a certain set point. So it will be raised to some degree, so we can consider that an initial positive feedback, but then beyond a certain value, there's still that certain set point that we don't want to go above. It's going to decrease, okay? So the answer is kind of yes and no. That initial part of that fever would definitely be a positive feedback, and then it would feed back into a negative feedback if temperatures get too high, right? That'll start to denature proteins, damage organs, and so on. But the intent is there, like the body is intending to do that, right? To attack that virus or bacteria initially at the outset of that inflammatory response. Yeah, that's a really good question though. Thanks for bringing that up. Okay. All right, let's save this and let's move on here. Okay, so the next thing we talked about were where we looked at enzymes, and just to kind of touch on enzymes here a little bit. These were essentially lowering the activation energy . So they decrease the activation energy of a reaction. In other words, make it more favorable for that reaction to occur. And then we can also say that they speed up the rate of the reaction, or they increase the rate of the reaction. Okay. We talked about a couple of things that increase the rate of enzymatic activity. So how fast that enzyme works. So factors that increase the rate of enzymatic activity . One is going to be the catalytic activity of each enzyme. Two, we said, was the substrate enzyme concentration. So both the concentration of the substrate as well as the enzyme. So substrate and enzyme concentrations. Right, if we have more of either of these, the rate of the enzymatic activity goes faster, or there, right? Substrate and enzyme concentrations. The third factor was the temperature and pH. Okay, those could also help to increase the thermal energy in particles and would further increase the rate of enzymatic activity. And then fourth was going to be the affinity of the enzyme for the substrate. Right, how attracted those two are to each other will definitely affect the enzymatic activity as well. Okay, the third thing that we sort of reviewed here that's important is the difference between endothermic and exothermic. So let's just kind of review that here. We have endothermic or endergonic reactions. So endothermic or endergonic. And that means that this is a reaction that requires energy. In other words, it is catabolic. An example here was making proteins from amino acids. Okay. Another way to express this is that we have more energy in the products. Okay. So more energy stored in the products. On the other hand, we have exothermic or exergonic reactions. Exothermic or exergonic. And this was the opposite, right? These donate or liberate energy. We can think about them as being, oops, I mixed this up. This is going to be anabolic, excuse me. And then this is catabolic. Okay. So catabolic means that we're now liberating or donating energy. Again, think about that positive or the T in catabolic. Think about the X in exergonic, the X in exothermic. That's just a way to think about the positive or the liberating factor of energy here. And then here we have an example of breaking down proteins to make amino acids. Right, just the opposite. So breaking down those proteins to amino acids. And then again, another way to express this is that we have more energy in the reactants than the products. Okay, so really important to understand these two different types of reactions, okay, that we talked about initially, but that we've also continued to speak about with relevant examples as we think about passive, active, transport, and so on, okay? All right, again, I'll move on here if there are no questions. Great. The next thing we got into were our biomolecules. So I'll kind of touch on those quickly here. So biomolecules. All right, these were made of several different molecules. So we had carbohydrates, not carbohydrates, excuse me, we had carbon, we had hydrogen, nitrogen, and oxygen for the most part, but we could also have other things like sulfur, etc. But these are the main components or the main elements of our biomolecules. And so these in different proportions would form our four main biomolecules. So lipids, carbohydrates, nucleic acids, and our proteins. Okay, lipids being really important in terms of their polarity, right? The fact that they are non-polar. Another way to say non-polar is that they do not interact nicely with water. Another way to say that is that they are hydrophobic, right? So make sure you are comfortable with different ways of expressing polarity. All right, we can say hydrophobic, we can say lipophilic, right? Those are different ways to express this idea of being nonpolar. So nonpolar does not interact nicely with water, hydrophobic, or lipophilic, right? Liking fats or being afraid of water, okay? And one of the main or most important, I won't go over all of the details of lipids, right? That'll be a bit redundant here, but the most important take-home part about lipids was phospholipids, and specifically the way that they contribute this amphipathic nature to the cell membrane. So phospholipids being amphipathic. Make sure you understand what amphipathic means. Right, having that polar head, but then the non-polar tails. And so this is going to contribute, very important for our cell membranes. Okay. Carbs, nucleic acids, proteins, these are all polar, right? We spoke about a couple of different properties about carbohydrates, the different types, and make sure you're comfortable knowing what's a monosaccharide, what's a disaccharide, what are the examples that we talked about. Again, I'm not going to go over them in detail here, but understand the difference between a monosaccharide, disaccharide, polysaccharide, and then the examples. Nucleic acids, again, same thing. Make sure you understand the examples, and then proteins as well. Proteins, we talked about a couple different layers to their structure. So we talked about peptide bonds, right? We also spoke about their secondary structure. Their tertiary structure, and then their quaternary structure as well, okay? So make sure you are comfortable with each of these. And then as far as generally understanding that these are all polar molecules, right? They all interact nicely with water, whereas fats do not, okay? All right, I'll pause here and see what questions we have, and then we'll move on to some other topics. Again, if you've got specific questions, you can let me know if there's specific topics that I'm not including here, or if you want me to add more clarity, feel free to stop me or just put it in the chat, okay? The fifth thing we went through was cellular respiration. And again, not going to go into too much of the details here with the interest of time, but I will touch on what's important for you to think about how comfortable you are with this in terms of doing well in your exam. So cellular respiration, we describe this as being the process of harnessing energy from our biomolecules. So harnessing energy from biomolecules to synthesize ATP, right? And remember, ATP was only made to be broken. Synthesize ATP, and then put a second point here that ATP is made to be broken to do cellular work. Right? We then spoke about the stages of cellular respiration, right? The various processes involved. Number one was glycolysis. And again, just kind of summarizing the main point of glycolysis here. First of all, it takes place in the cytosol. Okay. Second of all, the products of glycolysis are important. So what do we get out of glycolysis? So 2 net ATP, right? We also get NADH, carrying electrons there. We get pyruvate. Okay. And then because we split this off into 2, we end up having 2 pyruvate here, right? Beyond that last set of steps in glycolysis, it becomes 2. And so we end up with really 4 ATP, but 2 net, and then 2 pyruvate at the end as well, okay? This happens via substrate level phosphorylation. So let's put in a different color here, that the process by which we make these 2 net ATP is substrate level phosphorylation. Okay, let's look at the second sort of intermediary step here, the linking step. So the linking step, we said that takes place in the mitochondria. Okay. It's gonna help to produce acetyl-CoA and then carbon dioxide as a byproduct. So those are the things that we get out at the end of that linking step. But remember, this product here, the product of glycolysis, is what is shuttled into the linking step. Okay, I don't want to confuse you and put pyruvate here, but understand that pyruvate goes to the linking step, and then what we get out at the end is acetyl-CoA and then carbon dioxide as a byproduct. Okay, no ATP is produced in this linking step, really important. Then we get into the Krebs cycle. So step 3 here, the Krebs cycle, also called the Tca cycle or the citric acid cycle, a couple of different terms for this step here. And this is a cyclical series of enzymatic reactions. So not like glycolysis that was linear, this was cyclical. So the substrate was acetyl-CoA coming from the linking step. The product was also acetyl-CoA. That makes this a cyclical process or a cycle. Okay, this happens in the mitochondria as well. And when we say in the mitochondria here, we're speaking about in the matrix, right? Remember, we're not speaking about that across the membrane where the final stage takes place, right? That's going to be the electron transport chain. When we say mitochondria here, all of this means just in the center of the mitochondria, the matrix. So the mitochondria, the products that we get out at the end of the Krebs cycle are 2 ATP, right, per acetyl-CoA that enters. We also get NADH. We also get another electron shuttler here, FADH2. And then we also get some more CO2 as a byproduct. Okay. Let's look at the fourth and final step, the most important step. So if you don't understand any of these processes, make sure you understand the electron transport chain, right? You should understand each of those, but if you want to focus on one, focus on the electron transport chain. It's the most meaningful step of cellular respiration. This occurs across the inner mitochondrial membrane. Right, very specific location here across the inner mitochondrial membrane. It's going to yield 34 net ATP, right? So remember we got 38 ATP total per molecule of glucose. We got 34 coming from the ETC, 2 coming from the Krebs cycle, 2 coming from glycolysis. So that's where we get that total 38 for 1 molecule of glucose from. So we got 34 net ATP being produced here. And then we also have water as a byproduct, right? So when we convert that oxygen, when it shuttles, accepts those final electrons, we also get water as a byproduct, okay? And then we also have the oxidized form of our carriers here. So I'll just put oxidized form of our carriers. Right? Okay. Also important to note here that oxygen is standing as that final electron acceptor. So the function of oxygen is to act as the final electron acceptor. In other words, no oxygen, no movement of this chain, no ATP production, okay? All right, I'll pause here again and see what questions we had specifically about cellular respiration, and then we'll get into our driving forces, which is the next thing we talked about. Okay, so getting into next thing here, which were our driving forces. right? This is where we began speaking about membranes. I'm not going to review what we drew out in terms of the structure of the cell membrane, so make sure you go back over that and you're comfortable in terms of each of the structures, especially the phospholipids, and how they contribute to the fluidity, flexibility of the membrane. But then the next thing we spoke about after that were our driving forces and really how we move particles across these membranes. So driving forces. And so a couple of important terms in terms of understanding the driving forces. One was the membrane potential. And another way to express this is that it's the unequal distribution of charge across the membrane. Okay, or that separation of charge. So the unequal distribution of charge. In other words, positive and negative ions, right? Unequal distribution of charge across the membrane. That's really what the membrane potential is referring to. In terms of the value, we said it's usually about negative 70 millivolts in a resting cell. And what this negative 70 means is that there's an excess of negatively charged particles inside the cell with respect to the outside of the cell. Okay, just to reflect on what that negative 70 means. The other important term is the equilibrium potential. Excuse me, the equilibrium potential. And I'm going over these because I usually find students who don't do well in that first exam are usually tripped up by the terminology. And really, if you can't understand the question, even if you understand the topic, you won't really be able to answer it, right? So being able to comfortably discern these terms is going to help you understand the question, therefore answer the questions. Okay, what I've seen historically is that a student got confused between what the membrane potential was, what the equilibrium potential was. And so reviewing these terms and what they mean is really going to help you tie it all together in terms of answering questions, even though you understand like what those driving forces are. Really understanding the terms is important too. So the equilibrium potential, we said, is determined by the concentration gradient. Okay. And it's really the membrane potential for an ion. So it's the membrane potential specific to an ion, to each ion rather, at which the chemical and electrical forces are equal and opposite. Are equal but in opposite directions. Okay. All right, an example here would have been the equilibrium potential of potassium, which is negative 94, which means that when the cell has a membrane potential of negative 94, potassium is no longer moving, right? Potassium's chemical force is equal and opposite to its electrical force, okay? Then we talked about the chemical driving force, right? The chemical driving force really based on the concentration gradient. Always moves from high to low concentration. Okay, and then we talked about the electrical driving force, really based upon two things here. So one is going to be the charge of the particle. And two is going to be the polarity of the cell, that excess positive and negative charge, essentially the membrane potential, the fact that there's more negative inside the cell, more positive outside. And so the polarity of the cell is the second factor that influences the electrical driving force. Okay. And I don't have space to write it here, but when we think about the charge of the particle, we want to think about not only the type of charge, positive or negative, but the number of charge as well. Okay, so you can feel free to make that note in your own notes that we're thinking about not only the type of charge, but the number of charges, monovalent versus divalent, etc. And then putting that all together, we then come to the ECDF, which is the electrochemical driving force. And it's really just a sum of both the chemical and electrical forces. Okay. All right. Any questions so far? Any questions on that bit so far? All right. What we'll review here at the bottom is what happens, how we determine the forces acting in opposite directions. I won't go over what happens when they're in the same direction because that's pretty straightforward. We said if electrical is in one direction, chemical is also in that direction, the ECDF simply follows that direction. What we'll talk about here is what happens if the forces are in opposite directions. Okay, and what we will now compare is the membrane potential at that time versus the equilibrium potential of that ion in question. Okay, and based upon that, we had three different scenarios, if you will. Okay, so if the membrane potential is greater than the equilibrium potential for that ion, then the ECDF favors the electrical driving force. Favors the direction of, let's say, favors the direction of the electrical driving force, okay? If the membrane potential equal to the equilibrium potential, then the ECDF is 0, right? In other words, no movement, the particle is at equilibrium. Okay, and then on the other end of the spectrum here, if the equilibrium potential is greater than the membrane potential, then the ECDF favors the chemical driving force, or favors the direction of the chemical driving force, okay? All right. So that was pretty much the takeaway from what we spoke about as far as driving forces, what happens based upon the concentration gradient, what happens based upon the polarity of the cell and the charge on the particle, and then putting it all together, what happens as far as the movement, the general movement of the said particle, given that it's got two different forces acting on it. All right. The last thing we'll speak about here is the very final thing we spoke about, which was movement across our membranes, okay? So essentially the movement across our membranes and the different types of transport. And I asked you all at the very end there to think about that table that summarized the different types of transport and be able to recreate such a table. So this is what that table should ultimately look like. So we've got passive transport. Couple of things that describes this: spontaneous, downhill, okay, donates or liberates energy. And then we've got active transport. Right, and then again, some descriptive terms for this was non-spontaneous, uphill requires energy. Okay. So that's kind of our main, two main categories here. We then spoke about different subdivisions or different types of passive transport here. So let's kind of go over those at the bottom here. So the first one is that we had simple diffusion, facilitated diffusion, and channels as examples of passive transport. So let's kind of give these each their own column here, if you will. What am I doing? Okay. So we had simple diffusion, facilitated diffusion, and then carriers. As far as active transport, we had primary and secondary active transport. So two main divisions here with a couple examples. Okay. So let's start by reviewing simple diffusion and some of the important parts of simple diffusion. So first of all, we said it was always based upon the concentration gradient. All right, it's going to be determined, the weight is going to be determined by a couple of factors. So the weight of simple diffusion is determined by one, the magnitude of the driving force. In other words, the magnitude of the concentration gradient. That's another way to say that, right? The driving force is the concentration gradient, and depending on how big that gradient is, that will determine the weight of simple diffusion. So the magnitude of the concentration gradient. The other was the exposed surface area. So the surface area exposed for transfer. And then the third was the membrane permeability. And I won't go into the further divisions of membrane permeability, but remember things like size, charge, the bulkiness of the membrane. There were different factors. So we can have factors of the molecule that increase the, so I'll put here, we had factors of the particle, and then we also had factors of the membrane. Right, so make sure you go back over and think about which of those factors were of the particle, which of those factors were of the membrane. Again, in the interest of time and space, I won't kind of outline that here. The second thing was facilitated diffusion or diffusion through carriers. All right, this required a transmembrane protein. Also called a carrier. This still obeys the concentration gradient. So really this property here, always based on the concentration gradient, really applies for all three of these, okay? Although it's requiring a carrier, it's still going to obey the concentration gradient. Another thing about facilitated diffusion that really sets it apart from ion channels more so was that it is actively involved in transport. So it's going to play a role in terms of that configurational change, and it's going to be actively involved in facilitating transport. Right, we'll see with channels, they're not so actively involved in that transportation, okay? So the last thing here, channels, and remember we have many different types of channels. Most common are gonna be our ion channels. But the main thing here, these are also transmembrane proteins. Okay, They are basically passageways or pores. And then they are passively involved in transport, right? They're not going to do anything. They're just going to stand there and allow that molecule, that ion to go right through. So passively involved in transport. Okay. Some examples here, we spoke about aquaporins, which were water channels. We also spoke about ion channels, which are probably the more important of the two. Okay. Moving on to active transport here. So active transport, two main types. There are primary and then secondary. And so the main difference was where that energy that's required for active transport in general, where that energy comes from. From primary active transport, that energy is coming from the hydrolysis of ATP. So energy released from the hydrolysis of ATP. Really straightforward, right? And then the main example here was the sodium potassium pump. Okay. Secondary active transport, the energy here is released from passive movement of another molecule. So energy released from the passive movement of another molecule. Okay, the examples here we talked about were co-transport. Remember, co-transport, both molecules are moving in the same direction. That would have been the sodium linked glucose transporter, right? And that was co-transport. And then the other example we gave was the proton, the sodium linked proton pump, right? The proton pump. Oops. So the sodium linked proton pump. And that is basically counter transport, right? So hydrogen ions are going out of the cell, sodium is going into the cell. And so this is an example of the counter transport. Okay. All right, let me pause here. I see a question up here. Someone's asking, why do carriers change shape? Carriers change shape to really just spit the molecule, such as glucose, out on the other side. Okay, so it's not a passageway. It's not like a channel, right? It's different from a channel in that way. It's going to bind to one face of that carrier. Like, let's say glucose, for example, binds to one face of that GLUT4 transporter. The transporter shape shifts, and then it kind of spits glucose out on the internal surface of the cell. Or the external surface of the cell, because the transporter is going to be on the basolateral membrane. Okay. Does SI make any